welcome. It's great to have you, and we're looking forward to your talk. And without further ado, I will pass the baton over to you. And please check the chat for announcements and updates from Armin, and share your questions and the like, and then we can make sure that your questions get asked and answered towards the end of the talk. Okay, Sheila, I'll let you take it away.
All right, well, thank you so much for that introduction and for the invitation to come and talk to everybody today. I'm really excited about it. I have to warn you, I think a lot of you are going to find this talk pretty unsatisfying. And I think it's really because there are a lot of outstanding questions, and I think we shouldn't be satisfied. I would like to see us go beyond using BOLD and resting state functional connectivity as biomarkers to really getting to a place where we could start to interpret what they mean in terms of the underlying neurophysiology. But as you'll see, I don't really think we're there yet. And so what I've tried to do with this talk is just to kind of give a high-level overview of where I think this kind of this work stands, and hopefully stimulate a lot of discussion and a lot of ideas that for, how can we figure out more? How can we better isolate the components that we're interested in this signal? So I tried to keep the talk short. I'll try not to talk too long because I really want everybody to have plenty of time for questions and discussion at the end. There we go. Okay. So, if we think about the brain, we know that activity occurs at many different scales, right? Things that happen at the single synapse can be important for memories. We have, localized areas that process inputs from, say, the same eye or from a single whisker. We have different layers where you have stuff coming in maybe to layer 4 and outputs coming from other layers to go to different cortical areas. So there's a really complex structure to brain activity, ranging from, single synapses and millisecond time scale to kind of this whole brain activity that maybe is influenced by neuromodulation or sort of, the physiological states, and that might change over the course of minutes. Now, a lot of what we've studied in neuroscience are these neural circuits, things like, thalamocortical connections, where you can record from a few cells in the thalamus and a few cells in the cortex and try to figure out, how they're passing information back and forth. And then along with all of this, a lot of times when we talk about brain activity, we kind of think automatically of excitatory neurons, neurons specifically, but also sometimes specifically excitatory neurons. But there's also a lot of glial activity that goes on. And I think for fMRI, there's a lot of interest in astrocytes in particular and how they might work with the neurons and influence the MRI signal. Okay, so we know that the brain is complicated. We've got many scales of activity, and everything is getting mashed together into this resting state fMRI signal that we measure. So, from our millisecond to, I guess you could think of lifetime level of temporal changes, and our spatial scales of a synapse all the way to the whole brain, all of that information is condensed into these tens of thousands of voxels that we have with resting state MRI and the time courses from each of those. So with this all mashed together, is there any way that we can pull out specific components? And that's what I really want to talk about today. How can we get back to understanding what sorts of processes might underlie this resting state signal? And I think we have a hope of this because different things happen, at different spatial scales, at different frequencies, with different spatial distributions and with different timings. So if we take what we can learn about those processes and try to use that to extract information from the BOLD signal, it might be that we can do a better job of splitting up some of these components so we can understand more about what we see changes in when we start to go to, say, the patient population. So I want to flip perspectives now and talk about this from the perspective of what we can see in resting state fMRI. And of course, there's a lot of noise in resting state fMRI. We know that respiratory and cardiac cycles contribute to fluctuations in the brain. Motion is a big problem. We get scanner noise. We might have noise from the reconstruction that we use to put the images together, depending on what type of image acquisition we do. So I'm more or less going to ignore those for today. I mean, that's a big component, but it's not the only thing that's in the signal. There's a lot of work going on about how to get rid of those, how to clean them up, how to minimize them in the first place. But for now, we're just going to ignore it and make our lives a little bit easier. So other than that, we have what we call the global signal. And so maybe these are global activity fluctuations. I put a question mark there because I admit I'm a little bit skeptical at the moment about whether there actually is kind of this brain-wide fluctuation in activity level. I'll talk about that more in a minute. We have large-scale repeated patterns of activity. So these are spatiotemporal patterns that repeat throughout the scan. I'll show an example when we get to it. I'll also refer to these as QPPs, or quasi-periodic patterns. There's something we've done a lot of work with in my lab. And so you can kind of think of these as like stereotype changes from activity in one network to activity in another network, and where the timing and the spatial distribution of the activity is very similar across occurrences. We can have transient activation of functional networks. So you can imagine that, let's say maybe the somatomotor network is sometimes active or deactive relative to the other networks. That may be in a different timing than what we see with these large-scale patterns of network activity that we're calling the QPPs. And then there's localized neural activity. And a lot of times, this is what we're really interested in, this or transient activation of functional networks, at least from, say, the cognitive neuroscience standpoint. So we know that localized neural activity can contribute to the bold signal because it's the basis for the changes that we see in task-based fMRI. So, if you do a memory task, there are certain areas that increase their activity. So that's what I mean by localized neural activity. And we know that while you're in the scanner, these things are still going on. Your mind doesn't turn off just because you're laying on your back in a noisy MRI machine. So there's still changes going on that should contribute to the bold signal. I think whether we can actually localize those or not, or whether extract those, is still an open question. Right. So I told you I'm going to kind of go through these, I guess, one by one. And I want to start with the global signal. And I've highlighted in blue here the things that I think contribute to the global signal. Definitely there's a lot of noise in it. Again, ignoring that component, maybe there are global activity fluctuations. And I want to be very specific here about what I mean by global activity fluctuations. I mean something that changes at least throughout the whole cortex at a time. So the activity in the whole cortex goes up and down, maybe subcortical areas. I'm willing to, be a little agnostic about those right now because they haven't been as well studied. But at least cortex, everything goes up and down at once. So I don't mean, something where we see a selective change in one network compared to the other, because I'm going to call that more of these activation of functional networks, or QPPs. So I think that both of these types of changes, these QPPs and the transient activation of functional networks, should contribute to the global signal. Anytime you have widespread signal changes that are coherent, that coherent activation should increase the overall global signal, unless it's, completely offset by an equal deactivation of other areas. So you can see how these large-scale networks might kind of give rise to some of the global signal. And my question here is whether there is actual, global changes in activity above and beyond these components that come from transient activation of functional networks, or the QPPs. Now, global activity is really, quite a rabbit hole, and I'm not going to go too far down it. But I want to show you some of the data that is making me skeptical about whether there's really global activity. I'm not convinced that there's not, but I am wondering if there really is, or if we're just looking at, these contributions from widespread networks. So one thing from my lab, if you look at the top left corner, maybe you can see my cursor. If you look up here, we looked at which areas contribute the most to the global signal. And other people have done this as well. But what we found, of course, is that it's not evenly distributed. Visual cortex and somatomotor cortex tend to contribute heavily, frontal areas less so, very little contribution from subcortical regions. So to me, this doesn't really look global. And then there was another publication by a former lab member who was working with Hamid Haider at the time, who looked at the PET global signal during eyes open and eyes closed conditions, and found that the global signal went up. It was a shift to baseline, but it was really, again, driven by activity in those networks. And so down here, this letter C at the bottom left is showing the networks that have the strongest change between eyes open and eyes closed that drive the global signal. And again, we see these visual areas and things kind of in maybe somatosensory or motor regions, some of these areas here. It's definitely not evenly distributed. Okay, so for a long time, I was convinced that there was a global neural signal, or else maybe that it was just global noise. So one of the things that we looked at was if you have simultaneous measurements of local field potentials and fMRI, you can look at how the dynamics of one tracks the dynamics of the other. And so we did sliding window correlation between power and different frequency band from two electrodes and sliding window correlation from the bold signal from those same areas. And we did that with and without global signal regression for the bold signal. And what we found is that global regression increased the resemblance of the bold time courses to the local field potential time courses. So it did seem like global regression was taking out noise. Now, I don't think that's incompatible with the idea that global signal may also affect some large-scale brain activity. It may be that by taking out the global component, you're taking out kind of the large-scale influences and becoming more sensitive to localized neural activity. But I don't know yet if that's the case. And I've got some other data later that we can talk more about it. The other thing that I had really gotten excited about global signal with is a lot of the work from Tom Liu's lab, where they looked at global signal in eyes open versus eyes closed condition with and without caffeine and things like that. And we're finding that global signals seem to be related to vigilance levels. So that said, well, there's something neural in the global signal. But again, this could actually come from widespread network activity that's contributing to the global signal. And I think in support of that idea, there was a paper that came out in 2018 that showed that a vigilance sort of template based on which networks are most active during particular types of vigilance levels was better than the global signal in terms of predicting the features of EEG vigilance. And so that suggests to me that the global signal is actually kind of a secondary measure of these vigilance changes, and that we can get more sensitive to them by going directly to the networks. Okay, so I promised not to go too far down the global signal rabbit hole, and there it is. I'm going to stop there with global signal. And the other one I want to talk about is these large-scale patterns of activity, the QPPs that we see in the fMRI signal. And this it kind of interacts with this transient activation of functional networks. Because as I mentioned before, these QPPs are time patterns, basically, of network activity. And I'll show you what I mean. So the QPPs are mapped using a pattern finding algorithm that basically just takes chunks of the scan that are similar and averages them together to get a template. And so what we get out of the analysis algorithm is a pattern template, which is basically volumes over time, and the time is about 20 seconds for humans, that shows this complete cycle of activity that we're picking up. Along with that template, we get a correlation time course that tells us how strong that particular pattern is at any point in the scan. And so that's what we're going to be looking at when we talk about the QPPs. This is what one looks like. So this is based on Human Connectome Project data. You can see the anti-correlation in default mode and task positive areas, and this kind of stereotyped propagation along the cortex as activity switches between having one network dominant and the other network dominant. Areas in the cerebellum and subcortical areas are closely tied to the changes that we see in the cortex. And here we can zoom in on subcortical areas so you can see that a little better. So basically we've just changed the scale so it's a little more obvious, but you can see again the same sort of alternation of activity that lines up with the alternation of activity in the networks that we saw before in the cortex, and also involves some stereotype propagation in particular areas. One of the things I should mention is that, I just showed you the primary QPPs, the one that we find first whenever we do this sort of analysis. But if you minimize the contribution of that particular QPP, maybe by regressing it out, you can go on to find secondary QPPs, third QPPs, etc. Here I'm showing 6, but you notice they go from number 1 here at the top left through 3, and then 4 through 6 on the bottom. And you notice as we get to the higher numbers that they get noisier and noisier. And for each of these, the amount of variance that they explain goes down substantially. So how many does it make sense to really look at? I don't know exactly, but I'm guessing somewhere around 3 or 4. For the most part, we focused on the first 3 for reasons that you'll see. So I said that I wondered about how functional networks, relate to these QPPs. So if you make a standard functional connectivity matrix, and here we have, I think this is the class of parcellation sorted into the networks as described by Thomas Yao. So we have just the basic functional connectivity displayed as correlation between areas pairwise in the Human Connectome Project. And so each of these diagonal blocks is basically a network. And so these squares show intra-network correlation, and the off-diagonal squares show inter-network correlation or anti-correlation, for example, between task-positive network and default mode network. So if we take out QPPs, this is taking out the first QPP. So I'm showing the original correlation matrix on the top right, correlation that remains after we take these out on the bottom left. You can see we've already gotten rid of a lot of intra-network connectivity and a lot of the anti-correlation between networks just by taking out that main QPP. We can go ahead and take out, 3 QPPs, and by the time we take out 3, we are down to very limited correlation. And so, there's still some intra-network correlation, and I think that would be interesting to explore more deeply where those areas are. One feature that you definitely see are these diagonal lines within each block, and those represent connections between homologous areas in left and right cortex. So, it's showing that typical bilateral symmetry that we see in functional connectivity studies, but interestingly enough, it's not removed by the regression of the QPP like a lot of the other connectivity is. Now, that may just be because it's exceptionally strong, or it may be for other reasons that we don't really know yet. But if you look, a lot of the connection within these networks goes down to, close to 0. And so that implies that these QPPs are really accounting for a lot of resting state networks that we see in resting state fMRI. With that, we also looked at how QPPs line up with functional connectivity gradients. So this is a fairly recent way of analyzing the data described by Margulies et al. In 2016. And the idea is you basically, do a dimensionality reduction based on the similarity of functional connectivity profiles across areas. And so the first functional connectivity gradient looks exactly like a particular phase of the first QPP. And if you look at the dynamics of the QPP, it actually sweeps across the first functional connectivity gradient. And the same goes for the second and third. After that, things get a little bit less clear, partly because the QPPs tend to be so noisy. We think that QPPs probably account for these functional connectivity gradients too. Now, going back to global signal for just a minute, we had kind of wondered, we're like, well, are QPPs basically global signal? Is this what people are calling global signal? And it's not, because after we do global signal regression, the QPPs are still there. It shifts them a little. It basically shifts the baseline per subject. Some people, if you do QPP analysis on the individual level, show more correlation and less anti-correlation. But the greatest correlation is still in the areas that are high on, the ones that show anti-correlation. So it's more like a baseline shift than really any difference. And after global signal regression, these all become much more similar. So it really says to us that, global signal is not QPPs. It does not necessarily say, though, that QPPs don't contribute to global signal. So I think, how much of the QPP is picked up by the global signal is something that we're still kind of exploring. So where do these come from? I mentioned before that we do some microelectrode recording MRI studies, and one of them was to try to understand these QPPs. And so at the time, we were interested in infraslow electrical activity, meaning electrical activity in the same frequency band as the bold signal itself. So below 1 hertz for sure, and mostly below about 0.1 or 0.2 hertz. And we found that this electrical activity was very coherent with the bold signal and showed a high correlation to bold at a time lag that approximated the hemodynamic delay. So that made sense. It was bilateral, even when we looked at just the infraslow activity from one electrode, like this indicated by the arrow. So it seemed like, infraslow activity was a really good basis for some of these functional networks. Now, interestingly, at least under some anesthetics, where we have a fast vascular response, we were able to see sort of this biphasic activity that reminded us of the QPP. And so here you can see that, we have a strong correlation between infraslow activity and bold at the hemodynamic delay. And then we get kind of this anti-correlation, which kind of implies this sort of propagating signal like we see in the QPP. And so we made videos of this and looked, directly at the comparison between the QPP template that we obtained from the bold data alone, and then the correlation between one electrode in the brain and the MRI signal at different time lags. So this is what that looks like. This is the correlation between an electrode and every voxel in the brain at different time lags. And you see it starts negative, becomes positive in the medial areas, then lateral areas, and they sweep towards the medial areas. And then there's a following wave of these lateral areas, negative activation again. Now here's the QPP template from the same rat, from the same scan, without looking at that measured infraslow electrical activity. And so what you can see is that it's very similar. And we actually did the statistics and showed that, yes, it looks like these QPPs are picking up this pattern of infraslow propagation. So where does this come from? It looks like we've got a signature here. Here is something we can pull out of the fMRI data that might have a specific source. It's not localized activity, that's for sure. But we wondered, where does it come from? Why do you have this sort of repeated pattern? This comes up over and over through the scan. And so we came up with two ideas. One is that the brain has a structural network. And so you could maybe think of it as a resonance over the network structure. Maybe some sort of self-organization. So that's one idea. The other idea we had is that, well, there are some deep brain nuclei that have very widespread and pattern projections. And so maybe one of these deep brain nuclei was giving us some sort of pattern input that regularly kind of triggered this pattern of activity. So I'll talk about the first possibility first. So to look at whether the structure really could give rise to these QPPs, we made a brain network model following years of literature from many people, including Gustavo Deco and Olaf Sporns and Joanna Cabral. And basically what you do to build these brain network models is you need some DTI tractography or some other measure of connectivity between brain areas. In humans, it's usually DTI. In rodents, you can use other methods. So we use DTI to build a structural network. And so we estimate how strongly different brain regions are connected. And you have to parcellate the brain into a number of regions so that you can make these sort of discrete maps. Then at each node, you put a neural mass model of some kind. There are a lot of different ones. We've used a few common ones in the field. We let it run to simulate the brain activity that you would get across this network, and then we did QPP analysis. And what we found is that QPPs are kind of there, but kind of not. So what I'm showing in this graph is here in the top left is the empirical QPP pattern. And now to facilitate comparison with this model, we've summarized it by parcellating the brain into 60 some odd areas. And this is activity in that area over time. So this is kind of a condensed, flattened version of the QPP. But you can still see that anti-correlation, and you can see propagation along the cortex. So what we got with two different neural mass models was this sort of switching between two states. And it happened faster than what we saw for the QPP. So that, that switching between two states, that does look kind of like the QPP, but it's not nearly so complex as what we see in the real data. So, it says maybe there's a structural component to this, but it doesn't seem like that could account for, everything that we see. But I have to, include the caveat that the current network models that we used anyway, don't include physical adjacency. So they don't allow for propagation along the cortex, which is likely to be important for the QPP because we do see that sort of propagation in the template. Now, we did find that if we change some of the coupling parameters for these models, we could change the frequency of this activity. We still don't get the same complexity, but it does seem to have some dependence on the structural network. So another approach that we tried was instead of, trying to guess which patterns were the best, could we estimate, which parameterizations for these models were the best? Could we learn these model coefficients using a recurrent neural network? So we tried that. I'm not going to go into details now, but we can talk about it in discussion if you want. And we found that we were able to, basically learn a fit to these brain network models that gave us something that looked very close to our empirical activity. So here is the actual activity, and here is the estimated activity for two different models. And they look really good. We did this, we let the model run, and we did QPP analysis again, and we did better this time. So up here are a couple of examples. The top left two are empirical QPPs that we find. Over here at the right is what we'd gotten with the brain network model before, where we had kind of this walkie transition between active and deactive. And then down here on the left at the bottom are what we got out of this deep learning model. And it's starting to show some of the complexity that we expect with the QPPs that we see in real data. So maybe this is a promising way to go. So the other possibility that we had come up with was that this was related to some neuromodulatory activity. We chose to focus on the locus coeruleus, mostly for reasons of we have to start somewhere, and it seemed like a good choice. So we know that manipulating just about any of these neurotransmitter systems changes functional connectivity strength and how you see clustering in the brain. We know that these neuromodulatory systems can modulate arousal levels, and we had prior evidence that QPPs are different under different levels of anesthesia and whether you're, maybe really engaged in a task. So that made sense. The LC is known to be involved in task performance and trial-to-trial variability, just like people have shown with this anti-correlation between default mode network and task positive network that we see in the QPP. There are studies linking the LC to infraslow electrical activity in the cortex, which fits with what we had seen in rats. I think it's interesting that, the LC is the site of early alterations in some of these neurodegenerative diseases. And, so almost every one of these neurodegenerative diseases shows some sort of disruption in default mode activity. And so maybe it could be that you're losing some of that QPP organization that's showing up as this disorganization. And, kind of in a side study, one of our collaborators found that QPPs let us better classify Alzheimer's disease mice models versus control models because they did change. So it seemed like the locus coeruleus was a good target. And if we look at the QPP across areas, so these graphs are showing where the left posterior cingulate cortex has its dip, so its maximum deactivation, and where it has its peak, so its maximum activation. And so you see the cortex is kind of divided between, this is showing the number of voxels that have their peak at those times. So you see that the cortex is kind of divided into two sections, one that peaks when the default mode network is negative and one that peaks when it's active. But what I really want to focus in on right now are these voxels in the thalamus and brainstem. And so you can see that a lot of voxels in the thalamus and here in the brainstem peak earlier than the cortex, as if they might be driving this change that occurs in the pattern. And that, again, is consistent with one of these deep brain nuclei, maybe one involved in arousal, in driving these changes. So, we did some of the easy examinations first while we were trying to work out a good animal model. And so we looked to see if there were changes while somebody was doing a vigilance task. And we didn't really see any change in, how strong the QPPs were or how fast they occurred when somebody was doing this. But when we looked at how well they performed, we saw a close relationship between higher default mode activation, so that particular phase of the QPP, and a more variable reaction time. When the TPN part is dominant in the QPP and we gave stimuli there, we saw much tighter and faster reaction time. And that, again, is consistent with things that people had seen before looking at these networks. So that, was encouraging. So as an update, I just didn't get it onto this slide yet, but in some HCP data, when we looked at a harder task or something that requires more continuous performance anyway, we did see that the QPPs are stronger and more frequent than they are at rest. Again, consistent with them, reflecting some sort of increased arousal as a person does this task. Okay, so then we got to the rat study, and we started out with a pretty harsh study, and we gave rats DSP-4, which is a selective neurotoxin for the locus coeruleus. And so you give that, you wait about a week or 10 days, and at that point, the LC activity is down to something like 10 to 20% of what it is normally. And what we found is that when we imaged these rats after DSP-4, we couldn't detect the QPPs anymore. So if you see these patterns over here, we don't really get much spatial coherence any longer. It's just kind of noisy. It was the same sort of thing that we got if we ran our algorithm on a dead rat. So that, again, suggests that maybe these neuromodulatory centers are somehow driving these large-scale patterns that we see. So kind of to summarize this QPP session, QPPs could arise from neuromodulation or structural resonance. I think we have, evidence that's kind of inconclusive there, but we think that they account for most of the functional network activation we see, and definitely the functional connectivity gradients. They're more or less independent of the global signal, although they may contribute to it because they're very strong and very widespread. So that leads me to this last piece, and that's the idea of, can we detect localized neural activity? And maybe it should be transient activation of functional networks. Maybe we can't see, kind of a single area activate, but maybe we're going to see some sort of coordinated activation. And I think that's, a question that we're all very interested in. We know that this activity is in there because it's the kind of thing that we pull out with task-related fMRI. But can we detect it in the resting state? I don't know. So we were thinking about this, and it seemed like maybe the way to go about this sort of activity was to look at coactivation patterns. So we did. This is the idea that if you have resting state fMRI and you're looking at the time course, the high amplitude events really drive the correlation between two areas. So, maybe we see that left and right somatosensory cortex are always correlated, but maybe that's really driven by something like, 5% of the images from the scan where they have very high amplitude at the same time. So this is based on, the original work by Liu and Dunn back in 2013. And it has some similarities because to make these coactivation patterns very clear, they identify and average similar events, but they don't keep the timing information like we do for the QPP. So these are maybe like a snapshot of a QPP. So analysis, again, on the data where we had simultaneous LFP recording and fMRI. And we did a couple of different things. We did the standard CAPS analysis based on the bold alone. And then we did an analysis where we triggered on high events in the LFP signal rather than in the bold signal. And then average frames, a hemodynamics delay out. And what we found is that the coactivation patterns look very similar, whether we looked at the ones triggered by the local field potentials are the ones based on bold alone. So in this figure, I'm showing two different anesthetics. The arrow indicates the electrode. And so this first column is looking at LFP and bold correlation. And the next column is LFP-based coactivation patterns. And they look very similar to the LFP bold correlation. So it seems like for both bold and LFPs, these high amplitude events are really driving the correlation that we observe. And they're a small fraction of the data that drives the results. So Xiaodi was working on this, and he did kind of a different perspective to get another view of what was going on. And for this analysis, he triggered on, the bold, high amplitude bold events, or I should say different amplitudes of bold events, and then looked at the window before to see what was happening with the LFP. So he averaged across, all of the bold events that fell within the 90th to 100th percentile for strength, and got one response for each of these different LFP bands, and then went to the next 10% of bold events, et cetera, et cetera. So just looking at the broadband, what you can see is that for the highest bold events, the 90th to 100th percentile, the top 10% in amplitude, we see a really strong LFP response at about I shouldn't say response. We see a really strong change in the LFP at about 4 seconds before, which matches up with the hemodynamic delay. Now, when you get to, the middle percentiles, this becomes indistinguishable from noise. And then if we have a very low bold response, we also tend to see a low LFP signal. So it's only these high amplitude signals that really preserve the relationship between bold and LFP. So I really like to put up this graph. So this, we wanted to look at this further. So Xiaodi plotted every time point, the bold response versus the normalized LFP power. And again, just looking at this broadband graph here, we've done it for different bands, but the differences are not all that interesting, especially for isoflurane. Just looking at this broadband here, the top 10% of bold responses are given in this kind of orange color. And you can see that the LFP events preceding those could be stronger than normal, yes, but they could also be weaker than normal. So they could be more negative. So this is just a way to see what this correlation actually looks like. There's immense variability, even when we have a significant correlation. And so I think we really need to think about, how to extract information from this reliably. Okay, so I mentioned that I think CAPs are kind of a snapshot of what we see in the QPPs. And so Eric in the lab has gone through and first did CAPS analysis on data from the HCP and found different states. One was default mode dominant, and the others were kind of variations on TPN dominant. And then he did the QPP analysis at the same time. And then for each time point in the QPP, he plotted the likelihood that a certain CAP state was present. And so, at the edges where there's not a lot of differentiation, you don't see a lot of differentiation in the networks. At this point, where the default mode becomes dominant, you see a huge increase in the state where the default mode is dominant. And as we go through the switch, things kind of come back to baseline. And then when we have the task positive network dominant, we see that state that was dominant before goes almost to zero, and these other networks become more dominant. So it does look like maybe the QPPs are driving at least some of the coactivation patterns. Okay, so this is, I said this is going to be an unsatisfying talk, and this is why I think it's unsatisfying, because these are still outstanding questions as far as I can tell. First of all, I don't think we know if there really is a global neural signal, or if we just get QPPs or neural activity, network activity and some noise, and that makes the global signal. The reason I want to know this is because the global signal has been really interesting in terms of distinguishing between patient groups. There's some possibility that that's just because there are motion differences between patient groups, but I think that, I'd really like to understand what was going on with this. If there was a truly global neural signal, it might relate to something like, brain state, arousal levels, something like that. Or it may be that those effects just contribute to the global signal via these functional networks. I'd like would exist if there were no QPPs. I think maybe they would, looking at what we have left after we do QPP regression. And I think looking at, some further analysis of how those coactivation patterns line up with the QPPs might also help us to decipher this a little bit. I would like to know where these high interhemispheric correlations come from. Why are they so strong in the first place? And why do they stay after we get rid of the QPPs when most everything else goes away? One of the ideas that has been tossed out there is maybe these are some sort of, like vascular amplification. And that's why they're so heavily bilateral. And then I think the really big question is, can we detect localized neural activity beyond what we see with the QPPs or the network activity and noise? I think that's still very open. Right now, coactivation patterns at least look like they're partly driven by QPPs. I don't think, that's a very new analysis, and we haven't figured out the extent to which there might be other contributing factors yet. But I think that's very open and also very relevant for the field of resting state fMRI. So I just wanted to put in one slide to show you what we're working on now. We're trying to get a method going where we can validate what we see in fMRI against optical imaging, which is exciting because with optical imaging, you can directly label inhibitory neurons, for example, or excitatory neurons and look at those currents. So for example, we could take the bold signal that we acquire at the same time with this optical imaging, use the machine learning approach that I described earlier to estimate these latent variables, and then try to validate those against what we had measured simultaneously with optical imaging. So that is, coming along. We've got our optical and fMRI setup developed, and Wenju got a nice first image of the rat skull through our optical fiber bundle right here. So I think that's beautiful, and I'm really excited to see where that's going to go. I just want to put this slide up for resources. We have QPP code that anyone is welcome to have and use. We've got it as MATLAB. It's in the CPAT pipeline. And for the data, we mostly used HCP for the human stuff, but we have fMRI and LFP data that we're always happy to share. So I wanted to put up the acknowledgement slide and especially mention the people who really contributed to the pieces of this talk. The NAS has done a lot of work with the whole brain QPPs. Amrit did our brain network models and the machine learning method to estimate the latent variables from that. Zhaodi did the coactivation pattern analysis. Wenju has been involved in everything, but especially in all of our animal work, doing the simultaneous microelectrode recording and MRI and the new optical studies. And Eric has done the work that I was showing you at the end, where we were trying to relate these different dynamic measurements like QPPs and CAPS to understand how they come together. Now, this lab picture is old, but our new lab pictures look like this. And so it's hard to tell where anybody is here. But that's just the state of the world right now. So that's the end of my presentation, and I hope y'all have some questions and things to discuss.
Thanks, Shella. This is great. I like that old lab picture. Where is that? With the structure?
It was on the Beltline, but I think they took it down probably because people kept climbing on it.
Very cool. Okay, so just as a reminder to folks, oh, it's at the Beacon now, Angela Matt says.
Cool.
Yeah, in Grant Park. So just as a reminder, you can go to the Q&A section and put in your questions, and we'll be watching that as we go along. I'll start, I guess, with one question about your, when you did the removal of the QPPs and you showed that there was still some left-right, the homologous pattern. To what degree have you modeled, like work in our group with Ashkan Fageri, we've been looking kind of at more unified models of static and dynamic altogether, right? And so, kind of thinking of it as one thing. And I was just wondering if that could be signal that is maybe more constant, right, over the entire run, perhaps, right, less dynamic. Have you looked at that at all? I just was curious.
No, we haven't, and I'm really curious about that. I would guess that it's not. Just because when we look in rats, at least, if we look at left and right somatosensory cortex, again, it's still driven by those high amplitude events.
Okay.
And so, that to me implies that it's not that it's a constant time locked signal. It's more of these coordinated bursts.
But I guess if it could be both, right? Potentially, because if it shows up in the high amplitude events,
it could be both.
That doesn't mean it doesn't show up in the low amplitude events equally well, right?
Right. Yeah.
Yeah.
And part of it is.
Yeah.
Yeah, I think part of it's noise, right? How far these things could be constantly talking at a low level, and they're below our noise floor somehow, and we only pull out the burst. That's totally possible.
Yeah. There was one question from Peter Heller, which said, can you describe your successful machine learning approach in more detail?
I will do my best. This is really Amrit's work. Machine learning is new to my lab, so we've been kind of learning with him as he goes along. But what he did is he built an LSTM, so long short-term memory net, and he takes in a certain number of time points, I think something like, 50 time points from the resting state MRI data. And he set that up so that at each point, he makes an estimate of the hidden states. The hidden states here are the variables of the neural mass model that we put in. And so he estimates each of those for all of the areas that we've got with these weights that are already fixed based on the structural connection. And then he compares the output of that brain network model. He basically integrates a time step and compares it to the next time step that we get from the resting state fMRI. And he uses the error then as a feedback to learn those weights for the model itself. So we're kind of learning the parameters of our neural mass model. He's done a few different ones. We did the firing rate model as a very simple one. We did Wilson-Cohen because that is commonly used and has the excitatory and inhibitory contributions. And if you want more detail, I can try, or I can also put you in touch with Amrit.
Thank you. Another question I had was when you showed the relationship of, I guess it was the cortical QPPs to the thalamic ones and the delay differences. How do you control for like blood supply and hemodynamic delay differences, or did you in that context? Or
we did not. Okay. So we did not build in any explicit assumptions about hemodynamic differences. I know, people usually say that they're on the order of a second or so in the cortex. I don't know if anyone's certain about how that works for subcortical areas. In the rat, subcortical areas seem to be fairly similar to cortical areas from our work anyway.
Okay. I mean, when we looked at it in human data and across the whole brain, the delays can be up to 4 seconds in the hemodynamics or more. So that's hemodynamics, right? So that's right. Obviously not the source of the signal necessarily. So yeah, it's interesting, but there's a lot of information in the delay, I think, too, right? Yeah. So it's a very, like you said, it's unsolved but interesting, right? So
exactly.
Another question from Bennett Faber is, would it be interesting to try to get resting state data from sleeping subjects to compare with awake data?
Absolutely. I mean, I would love to look at that. We have a collaborator who's doing some of those studies, and I think we just kind of ran out of man to actually look at what's going on there. But if you want to do it, I'll send you some QPP codes.
Absolutely. Yeah, we've looked at that a little bit with some Enzo Tagliazucci's data, where they have people that fell asleep, and we do sleep tracking, right, of prediction of sleep stage. It's got EEG data as well, and they've got some papers on that as well. So I think there's already some work out there. So yes, definitely very interesting question to look at in that context. Let's see. I posted in the chat, you may have already seen, but there's a paper by Robin Miller from our group, which is looking at sort of whole brain polarization in bold fMRI data. And we found a lot of really interesting things. And we also looked at relating that to transient states. And we looked at it in the context of schizophrenia and healthy individuals, and in terms of like how long these itineraries of global polarization were, and they were quite different, for the two groups. So it seemed relevant and related to the kinds of things that you were showing earlier. So I just wanted to mention that in case you hadn't seen it.
Absolutely, thank you. That's great.
Yeah. And Robin, of course, and can reach out to her if you'd like. So, other questions? Armin, do you have any questions?
I just had a comment. First of all, thank you so much. It's very interesting work. I have been following your work with QPP for a long time. So if I understand correctly, can we say your result may suggest that those global signal activity and changes comes from what you call QPP, simultaneous QPPs? Is it like, it can be a conclusion?
I don't feel comfortable saying that's a conclusion yet. I'm kind of leaning in that direction, partly because I haven't found a lot of evidence of a homogeneous global signal. It seems like what we see in the global signal tends to be linked to certain networks. Now, that doesn't mean that there's not, a true global signal in there, but I don't think we've figured out how to get to it yet.
So I'm just trying to understand the relationship between this QPP and like a neural activity. So we have those propagation wave that you mentioned in the cortex, then we have those neural activity. So we have like kind of functional hierarchy. So can we say that there is like we have a different level of the functional interaction? At one level could be this QPP, and the like lower very bottom is like those neural assembly, and probably this global signal is like on the top of the hierarchy somehow. Just a question.
Yeah, I mean, that's kind of how I've been envisioning it anyway. I don't know, again, for sure. I think that's why this is, such an interesting area to dig into, because we're still trying to figure this out. But it does seem like, you have localized activity, and at each scale, the activity contributes to the next higher scale, right? So, your local activity contributes to your network activity contributes to your whole brain activity. But I think it goes the other way too. And I think maybe your whole brain activity constrains your network activity, which constrains your local activity. And so trying to parse this, I think, is going to be really hard but fun.
Yeah.
Yeah, and we also have the relationship between structure and function at the same time.
Right.
Yeah. Great. Well, thank you. And Armin, did you want to mention anything about the Slack follow-up?
Yes, so we will continue for a week. If you have any question or comment, please share with us on Slack, and I will upload the video as soon as possible. It take probably a day, and we'll share it with everyone.
Okay.
Thank you.
So Shella, thank you again for joining us, and thanks everyone else for participating and being a part of this. And hopefully it's been interesting and informative for everyone. And you can always follow up and dig into the details of Shella's talk by checking out the video after it gets uploaded. So thanks again, Shella.
Thank you all so much. On Slack.
Great. Okay. All right. Bye-bye, everyone.
Bye-bye.